Hi all, this is a short webinar made by the MEESO team dealing with how to quantify mesopelagic micronekton and macroplankton resources. More specifically, it is based on the rationale behind and the experiences also made by the Norwegian Institute of Marine Research during the development of our current protocol for sampling of mesopelagic micronekton. So the ideas and concepts presented here reflects work done over several years, also as part of the IMR MEESO efforts. MEESO Work Package 2 has several deliverables in document form that deals with various aspects of sampling mesopelagic micronekton. These can be found at the MEESO webpage at meeso.org. meeso.org, and maybe worth looking into if you are interested in details or in further details or details about who in MEESO to contact if you have more specific questions. Currently there are 2 reports available on the MEESO webpage from Work Package 2, but we will update the pages to make more material available once it becomes available. So, how do we actually quantify the mesopelagic micronekton then? It's usually there are 2 main categories of methods we use. It's either acoustic or net-based, and it is a mixed bag of fruits, but currently the majority of works being published on biomass and abundance of mesopelagic nektonic biota, it tends to be based on acoustic data. Now, acoustic methods have some obvious advantages in that they are generally capable of mapping large volumes of water rapidly and at high resolution. The most common method of acoustic surveying depends on hull-mounted transducers and consists of going over an area or a transect and recording the acoustic output and then converting this output into some output of biological relevance. There are also acoustic methods that depend on submerged transducers, but I'm not going to go into details on the acoustics. I will rather focus on the actual capture of the mesopelagic organisms. What I will say about the acoustical methods is that almost regardless of what you do, you will need proper catch data in order to interpret what you see. We call this ground truthing, and it's needed because many organisms and objects may actually give echoes that appear similar. But again, these objects or organisms may have very different acoustic properties. As I mentioned, the most common acoustic method consists of going over an area, typically a transect, a line, and measuring the sum of reflected signal from all organisms below this transect. Then we make some assumption about how much sound an average individual reflects to arrive at the total number of individuals in our transect. And then to arrive at biomass on the transect line, we multiply by the average weight of an individual. Now, if we assume that what we measured on the transect is typical of the area, we can get to an area biomass by scaling up the number to account for the fraction of the total area covered by a transect. In sum, that is a lot of assumptions, but the assumptions about what is present and how big the average fish or organism is are 2 central ones, and getting these properties wrong can give a very wrong answer. Thus, the converting of acoustic signals into biomass can't actually be done without proper catches. Net catches and acoustics are not complementary methods, but net catches are actually a prerequisite for using the acoustic method. This is the rationale behind focusing this presentation on the net catches, and is also the reason why we at our group have concluded that the perceived problem of quantifying the mesopelagic micronekton actually comes down to proper sampling of the mesopelagic micronekton. I will be talking a lot about trawls here. So let me just start by broadly defining a trawl as anything that is pulled vertically or horizontally through the water, i.e. including different sorts of nets, fisheries, trawls, etc. I'll actually backtrack a little bit by saying that I don't think trawls are the only way of sampling mesopelagic micronekton. Back in the 70s, people, for instance, advocated using purse seines, which is not so much pulled through the water as it encloses a volume of water and then sieves it. But as far as I know, this method is not in use anymore, presumably for cost and logistical reasons. There are also other, more modern sampling methods, like say optical imagery and environmental DNA. These methods are exciting, but they're pretty far yet off from providing us with abundance estimates, so I will not comment much on them. The most important types of equipment currently in use to catch mesopelagic micronekton are therefore these trawls in my terminology, and they broadly come in two types when we refer to micronekton. We have the devices that are designed to filter water. These are basically scaled up plankton sampling devices and typically have relatively small opening areas and relatively fine mesh sizes. The second category in common use are the fisheries type trawls with graded meshes. These types of devices are actually designed for commercial catches, and for epipelagic species these work by herding in the organisms. In front of the trawl are huge meshes that are hardly suited to retain anything, but the presence of these meshes actually scares the fish inwards towards the center of the trawl, thus herding the fish towards the center of the net. Towards the end of the trawl, these meshes taper down into ever smaller meshes, and ultimately they retain the fishes that are herded all the way to the end of the trawl. From a commercial perspective, there is an obvious advantage in using this, in that you can put a trawl with an enormous opening into the water. As long as you have herding, this is in effect an enormous funnel into your cod end. If you don't have herding, for whatever reason, there is no particular advantage to using these trawls, as the filtered or fished volume becomes virtually impossible to assess. So at IMR we have gone for filtering devices, but with opening areas that is more comparable to those you typically find in small fisheries trawls or devices depending on herding. The opening area of the smallest trawl we use is compared above to some of the commonly used filtering nets, and in the following slides I will go through the rationale for why we have gone this particular way. It is impossible to talk about sampling of mesopelagic organisms without mentioning avoidance. And avoidance of these trawls or nets by mesopelagic organisms are believed to be a huge issue. And the presence of avoidance or avoidance of organisms from our capture equipment have been suspected to be a factor for a very long time. And it is also a long-held paradigm that larger and faster is better for getting a representative catch of larger organisms. So considerations of the effects of avoidance is one of the primary reasons why we at IMR have opted to go for large trawls or relatively large trawls. In addition to potentially being better at reducing the effects of avoidance, our large trawls also filter large volumes of water. It is obviously better if you aim at catching rare individuals, and I'll come a little bit back to that later on. On the left hand side you can see some examples of mesopelagic nekton that have occurred in the catches from our trawls. And on the right hand side there is a schematic putting the opening areas of our trawls in scale to some other commonly used equipment for sampling mesopelagic micronekton. The large grey outline represents the opening area of our so-called 280 MN trawl, and that's our largest trawl in current use. But the two black areas are then our smaller trawls, and inside those are drawn up the opening areas of some other common types of equipment. Even if avoidance of mesopelagic fish is perceived as extremely important, it hasn't received that much attention in literature. For plankton, studies have tried to use theoretical models and their parameterizations based on actual catches to assess how much of a bias there is in our catches. On the right-hand side, I've added a figure and a table from two often cited papers. The top one is an important theoretical model from the mid-60s that sparked several investigations back then. The important points of this work is that it used physically interpretable variables such as detection distance, avoidance speed, net speed, and opening to describe the mechanics of the avoidance process. The lower paper shown in the table and the title is a paper from 2004 that tried to estimate the influence of avoidance on our estimates of diel vertical migrations and biomass. In this case, the table shows that their studies suggest that the net catches underestimate the biomass, or biovolume in this case, of plankton by 10 to 20%, and that this error has an outsized effect on the vertical migrators. The avoidance behavior of krill, which in itself is an important part of the mesopelagic micronekton, has received quite a lot of attention. But while there has been a lot of talk about the uncertainty introduced by the avoidance behavior of the mesopelagic fish, there's been relatively few studies that have actually tried to quantify the avoidance. Two often cited examples that actually have attempted this quantification is listed above, along with a figure from one of them. So the figure is an echogram, which is the typical output you get from an echo sounder. So shows a time depth mapping of backscatter, with the amount of backscatter represented by colors. Overall, there is relatively little backscatter above approximately 150 meters depth. Apart from that, the main feature of the figure is this void in the backscatter, a U-shaped void. This white U-shaped void coincides with the vertical profile of a trawl. In other words, the figure demonstrates that where the trawl was, the mesopelagic organisms are no more. What the authors here did is that they compared what they got in the catch with what they estimated based on the lacking backscatter. And they then ended up with concluding that what is left in the cod end of the trawl was about 1 order of magnitude less than what was originally there, or in the water column. The other paper listed there came up with a largely similar magnitude in their estimate. Now, however, the weakness of both these studies is that they used fisheries-type trawls. Thus, they are in fact studying the effect of both avoidance and of fishes swimming or falling through the coarse meshes at the front of their devices, with no real way of separating what is what. And that brings me to the next important factor for sampling mesopelagic nekton, that is mesh extrusion. In other words, what is lost through the meshes after already entering the trawls or nets. There are probably other studies I could have used to highlight mesh escapement, but I really like this 2017 study from Alaska, where they put small fine meshed nets on the outside of their graded trawls to see what actually came through their meshes as they fished. You can see in the upper left hand corner an illustration for one of their trawls, showing the location of their pocket nets in several different positions around the trawl body. This figure is probably a little bit too busy, but I'll go through it anyway. In the upper right hand, the figures show that, unsurprisingly, what you get in your catches depend on which trawl you put in the water. This goes for both size distributions, which are the upper histograms, and also for the species distributions, which are the cake diagrams. Naturally, these effects have huge repercussions for how you interpret your acoustic data. That is, from which organisms the echoes did originate, but also the size distribution you use to convert the acoustical results to biomass. The lower left-hand figure shows the relative catchability curves the authors estimated for their trawls. And both curves show low catchability for small fishes with the relative catchability increasing at some intermediate size. That's the black lines. Now, it is worth reminding you that the species in this particular study were shallow living ones, and that the size distribution seen here may not be representative of that of mesopelagic fishes. The general consensus seems to be that mesopelagic fishes on average are small, which is arguably where you see the largest discrepancies between the trawls studied here. To exemplify this last point, I'll throw in another busy slide showing some data that IMR collected for cyclothony. As some of you might know, the cyclothony genus is believed to be the most abundant genus of fishes. However, many of these cyclothony are small and slender, meaning that they are very likely to be lost through any coarse meshed equipment. In the center here you can see an individual we observed in situ with an optical sensor. It's in the middle up there. The length of this fish is something less than 20 millimeter long. Some studies have, based on comparison between plankton nets and trawls, estimated that as much as 95% of the cyclothony catch is lost from their graded trawls. It follows that many nets give very poor estimates of both biomass and abundance of these. On the left-hand side of the slide is 3 histograms. And in the upper one, the green bars shows the size distribution of cyclothony caught in a typical plankton net with 180 micrometer mesh size. Note that all the units on this graph is related to abundance or mass in units per cubic meter. The black line shows the cumulative distribution of mass of these cyclothony caught in the plankton net. In the center, we show the catch distribution of cyclothony caught in our trawl. This trawl has a 3 by 3 millimeter mesh size along the entire trawl, but these results strongly suggest that even a 3 by 3 millimeter mesh size trawl does not retain the small cyclothony. They are simply lost through the mesh, and thus the abundance estimate and size distribution from our trawl is heavily biased. We can also see, however, that the trawl has higher densities of organisms in the larger size classes, and that for the macroplankton trawl catch, the average weight of a cyclothony would therefore be much higher than that which is found in the plankton net. So these two nets in this case simply give different data. In this case, we attribute the loss of small cyclothony in the trawl to the extrusion. And the absence of large cyclothony in the plankton net can either be an effect of too small sampled volumes, or it can be because of avoidance of cyclothony from the plankton net. In this concrete case, we did not try to estimate the catchability curves, but simply per length group we used the highest estimate in our final compound estimate, which is shown in the lower panel on the left-hand side. The interesting feature of this panel is that it is evident that the total abundance is dominated by the size fraction caught in the plankton net. But if you look at the cumulative biomass curve, about half of the total biomass of the population is in the size fraction that was not caught using this equipment. The right-hand figures in this slide I've included from a work we did in the North Atlantic some years ago, just to highlight that there are actual differences in the size distributions of mesopelagic organisms between areas, even in areas that are relatively close together. So the upper panel shows the size distribution, the cumulative size distribution of mesopelagic organisms, excluding gelatinous ones, in the Norwegian Sea, the Iceland Sea, the Irminger Sea, and the Labrador Sea. And panel B below there shows the normalized biomass spectra. At this point, I'll sum up the main arguments for using trawls or nets with the same mesh size along the entire body of the trawl. This, in the end, all comes down to herding. If you have a type of organism that can be successfully herded, a graded trawl can efficiently be used to increase the volume you sample. The downside is that once you have a resource that can either only be partially herded, or you do not know how herdable that organism is, the use of a graded trawl with tapering mesh sizes along the body leaves you with an effective capture area that will vary with both the sizes and the taxonomy of the resource you are capturing on. This gives increased bias in the catches, and also it is in practice impossible to accurately assign the effective capture area, meaning that these trawls are not really quantifiable. It might be possible to arrive at estimates of magnitude of relative biases using various techniques, such as for instance the pocket nets method mentioned earlier. But this is in practice very rarely done, and also the results may be very dependent on the local taxonomic size and state composition of the catches, raising doubt about whether this is a practical method to use for large-scale surveys, for instance. Thus, net catches and trawls are necessary for mesopelagic studies for the foreseeable future. But these trawls and nets can also be used as platforms for other instruments. At the Institute for Marine Research in Norway, we have spent a great deal of effort at improving and testing the sampling gear we use to quantify the mesopelagic. But I should also mention that we and other MEESO partners also have worked with improving our methods for monitoring the performance of the sampling equipment. As a result, we are now also using our trawls more or less as platforms for sending other instrumentation down into the mesopelagic. Most of these instruments give us some indication about the performance of our trawls, but the data we get off these extra sensors also give us some information beyond what we can get from the trawls, from the catches in the trawls. I should also mention that this is an old idea, and back in the day, mesopelagic researchers had high sensitivity light meters that they attached to the trawls to assess in situ light levels at the depth of capture. And this is something I wish we had today. Among the equipment we have routinely had on our trawls during the MEESO project, I would like to highlight the following systems. We have used autonomous ADCP systems along with other trawl monitoring systems to have a continuous record of trawl openings and flow into the trawls, enabling us to get a much better estimate of actual volumes filtered by our trawls. We have also used the deep vision system, which is a stereo camera system attached to the end of the trawl just before the cod end, to map taxonomy and sizes continuously during the trawl holes. For vertically structured systems, this is very important data, and we're still in the process of assessing the quality of them. Finally, several of the MEESO partners have been experimenting with putting autonomous echo sounders, such as Simrad's VBAT systems, for measurements either of organisms going into the trawl or looking forward ahead of the trawl. One of the things we wanted to do with the forward-looking echo sounder attached to the head rope of the trawl was to have a look at what organisms were doing in front of the trawl in order to try to quantify the avoidance behavior. This slide shows some figures from a paper we published in 2021, and in the upper left-hand corner is a schematic showing the attachment of the echo sounder to the trawl and also a picture of the autonomous echo sounder attached to the head rope of the trawl taken in situ. Next over is an example figure showing what we would expect to see on the echograms from this echo sounder. So we have drawn up a lot of lines approaching the transducer of the echo sounder, showing that organisms are coming in towards the opening of the trawl at an even speed. Now, if you have some avoidance behavior, we would expect the tracks to deviate from the typical line, which is defined by the trawl speed. So this is the schematic of what we expected to see. Now, we found out rapidly when deploying these echo sounders that they also give us a very usable metric of how many echoes we see in front of the trawl, giving us vertical abundance profiles. And the lower left-hand figure is showing such a vertical abundance profile divided into depth range and also the acoustic target strength or acoustic size class of these echoes, or acoustic strength of the individual echoes. On the right-hand corner, the count-based results from the trawl is also overlaid with the results we got concurrently with a hull-mounted echo sounder at a different frequency. And as you will see, if you compare the red to the black line, our count-based abundance estimates did not coincide with the strongest backscatter. So this is highlighting why submerged equipment is needed to study the mesopelagic. There might be a poor correlation between actual abundance and amount of backscatter. This is something we're working further with. We routinely deploy this echo sounder on our mesopelagic trawls, and here are another couple of echograms obtained from this forward-looking echo sounder on the trawl, showing individual mesopelagic organisms approaching the trawl. In both, you can see that many of the organisms apparently has no behavior relative to the trawl. In the left-hand figure, the typical approach speed is indicated with the orange line. But you can see in this echogram a couple of individual organisms that apparently do have some behavior in relation to the trawl and approach the trawl at much more slowly than the typical approach speed. The second echogram shows an aggregation approaching the trawl before vanishing some 15 to 25 meters ahead of the trawl, and individuals from the aggregation can be seen probably trying to avoid the trawl. I've picked out two reasonably clear examples here that I would interpret as avoidance, or at least containing some examples of avoidance. But as you can see, even in these echograms, many organisms appear not to avoid the approaching trawl, at least as we can detect it. So even with these direct observations of behavior, we're finding it hard to quantify avoidance properly, and we're continually looking for ways to improve our overall analysis. However, it seems to be hard to reconcile what we see on our direct estimates of behavior, or our direct observations of behavior in front of the trawl, with an order of magnitude drop in abundance in the catches. We're looking into both avoidance further away from the trawl and very close to the trawl opening, as well as the possibility of using the acoustic signal strength to look at whether avoidance is predominantly displayed by larger organisms. But as showed earlier for cyclothony, an order of magnitude difference in abundance and in biomass may be two different things, and I'll therefore spend a bit of time talking about the precision and accuracy of the trawl sampling. When we go out and do these mesopelagic studies, at least when we write them up and publish them, the precision and accuracy of our estimates are rarely in focus. In zooplankton studies, sampling error has received some attention over the years, and there have been studies looking at the effect of changing the hole length, net sizes, and factors like that on the sampling error. Of particular concern to most zooplankton studies are the effect of patchiness on the resulting estimates. So uneven distributions of the resource form the backdrop of many of these studies. With uneven distributions, overall precision and accuracy will tend to depend on sampled volumes. So one way of increasing precision is to increase the sampled values. The table shown on the left is from a simulation studies from the early 70s, and the values reported show that the coefficient of variation, which is a measure of sampling accuracy in these simulated catches, as a function of varying sample and distribution properties. As expected, increasing both net size and toe length decreases this coefficient of variation, but the details of how these change depend also on the properties of the distribution of organisms. We know that there are large vertical gradients in distribution for mesopelagic nekton. Horizontal patchiness has received far less attention so far, but there is good reason to suspect that many of the lessons learned about plankton sampling are more or less directly transferable to the sampling of mesopelagic nekton. The sample volumes also have implications for what we can actually use the data for. It's easy to make trivial examples to highlight this. We, for instance, don't go out and sample large jellyfish with the water bottles we deploy on our CTD probes. They're simply not suited for the job and sample way too small volumes of water to be used for that particular task. When it comes to mesopelagic fish, however, patterns are more subtle, but I'll try to clarify with a worked out example. Since we don't really know what the size distributions of mesopelagic fish are, I've taken the log normal distribution used in the modeling paper by Proud et al. From 2019. I've toyed a bit with the variance, letting it vary over a set of values. You can see the resultant length distributions in the lower left-hand corner, lower left-hand figure. In order to work with biomasses, these lengths must be converted. And in order to do that, I've opted to use the length weight regression for the Myctophid Myctophum punctatum as an example. So the central lower panel shows the resultant cumulative biomass curves for these different length distributions, plotted as a function of fish weight. The last piece of the calculations is turning these population size distributions into actual densities. I've done this simply by assuming that this hypothetical Myctophid population has a total weight of approximately 10 gram wet weight per square meter sea surface area. My reason for doing these off-the-cuff calculations is that I want to have a look at what sort of volumes one would need to sample in order to be able to characterize the population. As a very rough starting point, we can start with the inverse of density. If something has a density of 1 per cubic meter, the expected outcome of sampling 1 cubic meter is 1, and you can't sample fractional animals. So logic dictates that your sampling volume should be larger than 1 cubic meter. Applying the same logic, if you have something that has a density of 1 per 1,000 cubic meters, you can't really sample less than 1,000 cubic meters and still expect to get one animal. The final graph on the lower line shows the inverse of density for the fictional 10 gram wet weight per square meter population of Myctophids. As can be seen from the graph, we expect that very high sample volumes are needed in order to catch the larger components of the fictional population. And since this larger fraction of the population holds a rather large fraction of the total population biomass, the question can be raised whether small equipment is capable of proving valid biomass estimates. At this point, I have covered most of the points about sampling mesopelagic nekton that we in the Institute for Marine Research MEESO group believe are important. A large opening area of the collection device is important to reduce the effects of avoidance, improve precision in the estimates, and likely also to span the entire relevant size spectrum. At least if levels of biomass is of interest, this is very important. I have not covered the speed of retrieval or trawling much, but a faster net is likely to be less influenced by avoidance than a small one if all other factors are equal. Non-graded and mesh sizes attuned to the actual size distribution are important to reduce the amount of catch escaping through the meshes. It's hard to give an exact mesh size here, because it will depend on what is the target size range, and also what is possible, as the combination of mesh size and opening area will combine to impose drag. There are models and parameterizations of these models to estimate degree of retention for different mesh sizes and different sizes of fishes, which make these models a convenient starting point for these calculations. For the mesh we use on most of our trawls, which is 3mm bar or 3 by 3mm light opening, these models seem to concur reasonably well with our experiences. But even this very fine mesh size does not effectively retain all of the mesopelagic fishes we are aiming at. I have not covered the ability to collect depth stratified samples, and that is because we at the Institute of Marine Research presently don't have the capability to collect these samples in a quantitative way. We do have cod end samplers available for our trawls, such as the deep vision system and the multi-sampler systems, but a high proportion of the organisms entering the mouth of the trawl get tangled up in the mesh somewhere along the way, so that the trawl transit times is variable. This factor is also likely to vary between different species, as some species seem more prone to get hung up than others. Similar issues were identified in other cod end sampler systems as far back as the 60s and 70s. So it may simply be that systems where whole nets are fished instead are the way to go in order to get depth stratified samples. Apart from that, it's probably a good idea to construct nets and trawls in a way that makes them maximally stealthy, at least when quantification of the mesopelagic is the aim. For commercial catches, stimuli inducing herding behavior might be the way to go to optimize the catches. In the end, I don't think there is really a one-size-fits-all equipment for micronekton sampling. The IMR-MEESO group is using at least 3 different types of catch equipment to ensure that we can quantify abundance and biomasses over the size spanned by the mesopelagic micronekton. This slide summarizes some of the different quantitative trawls IMR has developed and tested over the years, and has some info on the ones currently in use. As I mentioned, they're all built on the same principle, as sieving rather than herding devices, and have an even mesh size from mouth to cod end. They also have minimal trawl wings and are deployed with constraining ropes at the mouth, which enables us to use large trawl doors that run such that the doors and warps are as far away as possible from the mouth of the trawl in order to minimize the herding effect of these components. To strengthen the trawls, we have an outer cover mesh on them. The largest trawl listed on the left was in practice simply too large for us to handle efficiently and is no longer in use, but the 3 other trawls constitute our current arsenal of quantitative micronekton sampling devices. We believe these trawls enable us to sample typical mesopelagic micronekton much more quantitatively than other types of equipment, while we still recognize that there is no perfect sampling equipment for all of these components. However, it is important that we use equipment that reduce the inherent bias in our current estimates. We are in the process of writing up a paper that summarizes the development process and testing of these trawls, which by now have occurred over quite a long time frame. For those interested in further reading on the topic of net catches of mesopelagic micronekton, there is some literature published on the topic, and I'd especially like to point to a special issue of Biological Oceanography from 1983, dedicated to the work of the SCORE Working Group 52 on micronekton from 1977. There are quite a lot of good articles in this volume. 2 of them are listed on this slide. Other than that, as I mentioned at the beginning, Work Package 2 of the MEESO project has also published synopsis of the work that occurred under the MEESO umbrella, and this is available via the MEESO webpage. To sum it up, net catches are a necessity on its own, but also for using acoustic methods for studying the mesopelagic. While we recognize that acoustic is an important and convenient tool for mapping the mesopelagic, we believe that in general, and as a general observation, current mesopelagic studies should put more effort into the sampling of the mesopelagic organisms.